Next, we'll be joined by two speakers who are speaking on behalf of OrphAI Therapeutics. We'll hear from both Dr. Suma Babu, who's co-director of Neurological Clinic Research Institute at MGH, and Dr. Brigitte Roberts, who's CEO at OrphAI Therapeutics, who are going to tell us about apilimod dimesylate development to treat ALS. Welcome.
Great. Thanks, Fernando. So good afternoon, everyone. I'm Brigitte Roberts, the CEO of OrphAI Therapeutics, and I just wanted to thank you for inviting us to present at today's conference. So OrphAI Therapeutics is a small biotechnology company based in New Haven, Connecticut, and we currently have 3 drugs in development for conditions really where there's a true unmet medical need. The topic of today's discussion will be AIT-101, which is the company's proprietary PIK5 kinase inhibitor in development for ALS. We delivered phase 2A biomarker-driven data last year, which our lead PI, Dr. Suma Babu, will be presenting here today. And we are now working very closely with Dr. Babu on the next stage of clinical development for the compound. So with that, I'll turn it to Suma.
Thank you, Brigitte. Good afternoon, everyone. Can you go to the next slide? So AIT-101, or apilimod dimesylate, is first-in-class, most potent selective PIK5 inhibitor. And this is the drug that we tested in our phase 2A trial in ALS, specifically looking at C9 or 72 positive ALS individuals as our first study to look at target engagement, as well as CNS penetration. We can go to the next slide. And so AIT-101 works on the lysosomal pathway. If there is PIK5 inhibition, there would be an upregulation of the TFEB transcription factor EB, which then would increase lysosomal gene expression. And if there is an increase in lysosomal gene expression, you would expect to see a couple of different downstream effects, including clearance of toxic aggregates within the cell. And in this particular case, we are focused on C9 individuals, so we'll be looking at aggregates, specifically dipeptide repeats or poly-GP in terms of clearance. And we're looking at a target engagement biomarker, which is the soluble GPNMB that we selected for this particular trial. So if there is lysosomal gene expression that occurs secondary to PIK5 inhibition, you would expect to see an increase in the biomarker GPNMB, which is a secreted protein from cell surface. It's a neuroprotective and anti-inflammatory protein. And so we'll talk about how the GPNMB was just covered later in the talk as well. So I'd like for you to focus on those 2 biomarkers for now, because we're going to talk about the results of this in a little bit. And as you can see in this pathway, neurofilament light actually doesn't show up anywhere along the direct pathway of PIK5 inhibition. But that's a question that's on everybody's mind in terms of any trial in ALS. What happens to NFL? We'll talk about that as well in terms of learning from this study. And all of the data that I'm presenting today is already published. It was published a few months ago in Brain, so even the supplement has extensive information there. Anybody who wants more detailed information, please refer to the Babu et al. Brain paper that was published. You can go to the next slide. So this is a clinical trial design. It was a very small study. Oops. Looks like somebody else is sharing their screen. Oh, thank you. Yeah, and so the beauty of this study is really about the very small sample study and biomarker-driven study. At this stage of drug development, what we were most interested is to understand, does this drug, when taken orally in a capsule formulation, cross the blood-brain barrier? Does it engage with the target in CNS? And do we see changes of target engagement in terms of protein aggregate clearance in the CNS? So that's sort of our main interest at this stage of development. We're not looking at clinical efficacy at this stage, and that will be the next stage that we're focusing on. And to answer this question well, I'm looking at safety, of course, in ALS population, because this is the first time that this is being tested in ALS population. So to answer these questions well, all we needed was 15 ALS participants, 10 on active and 5 on placebo, 3 months of randomized control trial design, followed by an open label extension. And that was the study design that we used here. And our primary outcomes, as I mentioned before, safety, tolerability, and CNS penetration of the drug, so measurable drug levels and active metabolite levels in the CSF. And our secondary endpoint was really the target engagement. So the GPNMB is an on-mechanism biomarker for PIK5 inhibition, and our poly-GP or dipeptide repeats are on-mechanism target engagement within the CNS in this disease-specific population. And so we did measure NFL. We also measure clinical measures, as well as lower down in the hierarchy as exploratory outcomes. We can go to the next slide. This was our cohort, and predominantly participants in both groups were slow progressing, longer duration. And the placebo cohort particularly was notably slow compared to the active, compared to the group that was randomized to the active treatment, as you can see here. Go to the next slide. And this is our overall consort diagram. So as you can see here, majority, almost everybody who were enrolled in the randomized control studies, a 3-month-long study, completed the randomized control portion. One dropped out early in the active treated group because of nausea that happened very early on. And otherwise, 9 out of 10 participants completed the randomized control portion in the active treated group, and all 5 in the placebo. And in between the randomized control and the open label extension, one early terminated because of COVID and other comorbidities that emerged there, which is unrelated to the drug itself. And then out of the 8 in the early treated and 5 in the delayed start group that entered the open label extension, 7 completed the completed week 24, and 3 completed, and the 3 dropouts were because of disease progression, difficulty to swallow, which was required because it's a capsule formulation, and respiratory failure. We can go to the next slide. But overall, really good retention and on-drug tolerability. So this is our first target engagement biomarker. What we saw is poly-GP, a dipeptide repeat, which is a widely used biomarker in C9 clinical trials as a target engagement biomarker, showed a 73% decline within 12 weeks. And I think this is almost comparable to some of the ASO trials that show target engagement to this extent. You have to keep in mind that AIT-101 actually works on post-translational mechanisms, so it's not really comparing it to other ASOs as such, but we saw a huge decline in poly-GP, which is favoring the hypothesis that there is CNS target engagement following the treatment of epilimumab mesylate. And these are with very, very small numbers. As you can see here, by week 12, we had 9 participants on active and 5 on placebo. Can go to the next slide. When we looked at the open label extension, so the delayed start group in red that then went on to the active treatment also showed a decline very similar to the early start group. And so the big takeaway here is that there was CNS target engagement in C9 individuals, which indicates that the drug crossed the blood-brain barrier and engaged with the target within the CNS. We can go to the next slide. So GPNMB as a marker of PIK5 inhibition also demonstrated an increase, as one would have expected. If there's PIK5 inhibition, there's an increase in TFEB, and increase in TFEB leads to increase in GPNMB. And we saw a statistically significant increase in GPNMB, 2.5-fold, and by week 12. We can go to the next slide. But on the other hand, CSF GPNMB did not show a difference. And then this raises a number of discussion points in terms of what do we know about some of these proteins in terms of their kinetics, and how they probably their kinetics differs in different cell types. And do we know, as I mentioned before, GPNMB is a protein that is actually secreted and cleaved from the surface of cells. And does it act the same way in CNS cells? Do they have the same metalloproteinases to actually cleave this? Or do we have enough concentration in the CSF to actually measure this? The CSF assays for GPNMB different. What we know is that the blood assay is the validated assay that we went after. So we don't fully understand how to interpret the CSF changes as such. In the next few slides, I'm actually going to show some NFL data, and this is a question that's on everybody's mind about what happened to NFL. But this has been a really good learning experience for us to actually look at how can we interpret NFL changes in very small sample size early phase trials. And can we really sort of hang our hat on what we see here? And I want to show in the next slide the huge discordance. You can go to the next slide, please. The huge discordance we saw between CSF and plasma NFL levels on an individual level. You can see that the directionality, the magnitude of change is really a variable between plasma and CSF. We've all looked at group level correlations between CSF and plasma NFL, and there's beautiful, excellent correlations. But when you go down to very small sample size studies like this, you see variability, and then you don't know what the net effect is going to be with all this variability. It's anybody's guess in terms of what the net group effect is going to be in CSF versus plasma. And it also raises, it was actually really a moment for us to pause and think about how much do we know about NFL? I think as a field, thanks to the tofersen trial, we understand really well how to interpret large decreases in NFL following treatment in trials. But do we really know how to interpret no difference or increase in NFL as a field? And I think these are things that we wanted to sort of think about, and this trial sort of gave us this opportunity to think about. So as I mentioned before, there's discordance, and so the net effect on a group level based off of this individual level discordance is anybody's guess. And what we ended up with, can go to the next slide, is that in CSF, there was no difference between active treated and placebo, and in the plasma, there was a slight increase in NFL compared to placebo. And so that raises all these questions of what do we know about NFL, kinetics of NFL? Do we know that, what's the source of NFL? Is it always in the CNS? And is it always a spillover from CSF into plasma? Or is there a separate peripheral source of NFL that could be contributing to some of this? And what do we know about NFL in C9 individuals, particularly C9 ALS? What's unique about C9 compared to other forms of sporadic ALS is that there are large anatomical regions in the brain, the frontal lobe, the temporal lobe, cerebellum, and these can can be involved in the disease at any point during the disease progression. And how do we account for NFL arising from these large anatomical regions as the disease evolves? And especially in trials, we're not doing a great job of monitoring the frontotemporal changes or cerebellar changes clinically or with outcomes in trials. So it's really difficult to interpret the net effect that we see in such really small studies, really comparing 9 versus 5. And I think that is sort of one important lesson for us to really pause and think in terms of incorporating NFLs in early phase studies, in small studies. There's also emerging evidence that there could be drugs that actually can induce NFL expression. There was a paper about minocycline recently. There's also another paper that talks about NFL increases in placebo group or decreases in placebo group as well. So I think these are all things that we have to sort of, we don't know the answers, but we have to pause and think deeply about it. You can go to the next slide. And in terms of clinical biomarkers, again, this is clinical outcomes. This is really an underpowered study for any clinical outcomes, and they are exploratory, and slow vital capacity is what you can see here, 7 versus 3 comparison at week 24. We can go to the next slide. ALS-FRS-R changes are shown here. You can go to the next slide. And this is the ALS-CBS, with, again, over small durations of time and small population. You can go to the next slide. So overall safety profile of AIT-101 was really good. And there were 2 serious adverse events. Neither of them were assessed as related to AIT-101. One pancreatitis, which was related as unrelated, which assessed as unrelated. There was one other drug treated individual in the non-Hodgkin's lymphoma trial who developed pancreatitis, who tolerated a drug rechallenge really well. And so probably this is unrelated to the drug itself. And then there was one death related to disease progression in the placebo group. We can go to the next slide. So GP and MB biomarker discovery, I think in terms of trial designs and really being thoughtful about the mechanism of action, the disease biology, and how do we select biomarkers that would show us the target engagement. And for this particular study, it was a thoughtful approach of actually proteomics in 12 participants, plasma samples from participants in the non-Hodgkin's lymphoma trials where they were treated with ipilimumab, looking at, in an unbiased assay, and it showed that 72 proteins directly sort of changed following apilimumab treatment, out of which 19 were significantly changed plasma proteins, and then all these 19 were validated, and GPNMB was selected because it was most upstream and directly related to the PIK5 pathway, and there was a validated blood assay that was available, and so that was a biomarker selected for the target engagement in this study. Go to the next slide. And it's always nice to then have another validated look at, we saw poly-GP decrease in participants in the trial, but do we see a similar decrease in poly-GP in patient-derived induced motor neurons? And we did. So this is almost sort of like showing biological efficacy in the Petri dish studies, reduction in poly-GP in C9 patient-derived induced motor neurons on the left. Go to the next slide. And this is the overall summary. So AIT-101, apilimumab dimesylate, is CNS penetrant, safe, and showed target engagement in C9 population by lowering the dipeptide repeats. And the next step of development of this drug is really to look broadly, because lysosomal pathway and protein aggregates are relevant to all forms of ALS. The next step is really to widen the study and the drug development to all forms of ALS, which we're working on currently. So the next slide. And with that, I would stop and acknowledge everybody who participated in the trial and all our collaborators who helped run the study at all 4 sites, and all the vendors as well. Thank you so much.
Thank you, Suma and Bridget. It was a fantastic talk. Thanks for sharing and updating us on the progress of this molecule. There's a lot of interest, a lot of questions, and we have some time to take them. So the first one relates to whether or not you measured any changes in LC3 or sequestosome as a function of treatment, because autophagosome is involved in this mechanism.
Great thought. We did not check in, we did not measure in this study, but certainly something for us to consider for the next phase of development.
And a follow-up question kind of relating to the correlation between age and neurofilament levels. Might there have been age effects and differences or discordance in the NFL levels that you were seeing?
That's another really important thought as well. So in this particular trial, age was balanced between the active and the treated group. So we don't fully understand. I think there's the plot is very deep when we look at non-specific biomarkers like NFL, but we don't think that it is really just age that is contributing to the discordance.
A question specifically about the GPNMB. What assay did you use to measure the soluble GPNMB, and what were the approximate limits of detection or lower limits of quantitation for the assay that you were using?
And that's a great question. And I'm a clinician. I would not know the details of this. I'm happy to bring this back and take it offline and circle back with whoever asked this question.
Thank you. A question about the NFL. Do you know if there's any evidence that NFL has binding proteins?
Oh, that's a really interesting question, and I don't know the answer to that. I don't. I would love to actually learn from others if somebody knows the answer to this, and I don't know the answer to this.
Thank you. A couple more comments here. One is a question regarding the selection of your dose. How did you determine the doses to be explored in your study, the dose to be explored first? And then I got a follow-on question that's kind of unrelated. Have you looked at soluble GPNMB in preclinical models?
Yeah, so the first question is that 125 milligrams twice a day was the dosage that was selected for this trial, and that's the dose that has been tested in multiple other apilimumab dimesylate trials, the non-Hodgkin's lymphoma trial, as well as the COVID-19 trial, and others. And there have been more than 700 participants who were treated with this particular dose, and we have excellent safety profile for that particular dose. And your second question was about soluble GPNMB and preclinical models. Yes, there was a spinal cord tissue that showed an increase in GPNMB following the treatment with epilimumab mesylate. All of this is available in the supplementary material of the brain paper that we've published. So if anyone's interested in looking at it.
Thank you. And just a comment to commend you for prioritizing the safety and PK and biomarker data over really looking for markers of efficacy in an early small clinical trial. There's a lot of enthusiasm for that approach. Thank you.
Thank you so much.